Would you like blue eyes with that? Or perhaps 30 IQ points? As disturbing as it sounds, questions like these could actually become a reality for expectant parents as gene editing technology progresses. Despite this unnerving future, gene editing also has the potential to save millions of lives. The decline or the salvation of society may lie in this technology. Good afternoon, my name is Owen Rancy, and these are my colleagues, Noah Park, Kadambari Bias, and Eleanor DeVorschfelder. And today we'll be analyzing the inquiry. To what scope can gene editing be utilized to cure otherwise untreatable genetic diseases while remaining moral and affordable to the average person?
Gene editing, first researched in the early 1970s, is the process of altering the genetic code of an organism. At first, there was a fundamental problem with this. Gene functions were almost completely uncharted. This meant that scientists did not even know which genes to edit. Two main techniques were used to solve this problem: forward and reverse genetics. Forward genetics starts with an irregular organism. Then the genes that cause this are found. Reverse genetics is basically the opposite of this. It starts with modifying the DNA base, then observing the physical impact on the organism. Through methods like these, the functions of genes were slowly but surely discovered. One such medical application is a recent technology called base editing. Researchers based at MIT and Harvard found that base editing can fix point mutations, or errors in single DNA base pairs. This allows for extremely precise cuts rather than just cutting out large chunks of DNA. Researchers based at MIT and Harvard found that there are currently around 16,000 known disease-causing point mutations. These could all potentially be fixed using base editing. There are many instances of those cured using gene editing. BBC News cites Matt Chappell, a participant in the world's first gene editing trials, who had his immune cells donated and edited to cure HIV. Additionally, doctors in Britain were able to cure a 1-year-old leukemia patient, Layla Richards, with donated immune cells. These examples are just the beginning of the potential that gene editing has in the medical field, but gene editing was still in its infancy. Since then, there have been numerous technological advances.
Thank you, Eleanor. There are many different methods of gene editing, such as forward and reverse editing, as Eleanor mentioned. However, the most commonly used method today is the clustered regularly interspaced short palindromic repeats, or CRISPR for short. CRISPR is utilized more frequently compared to previous gene editing technologies due to its greater accuracy. CRISPR is also increasing in popularity in the scientific field due to its ability to cure numerous genetic diseases, such as cystic fibrosis, sickle cell disease, some forms of blindness, severe combined immunodeficiency, some cancers, HIV, and many others. But how exactly does CRISPR work? According to the Massachusetts Institute of Technology, CRISPR works like a find and replace function. In other terms, there are three main components to this technology: the guide molecule, the healthy DNA copy, and the DNA cutting enzyme. The guide molecule pinpoints the target DNA strand, the DNA cutting enzyme cuts off the strand, and the defective DNA is replaced with the healthy DNA copy. CRISPR is also very versatile in nature, which means that it can adapt to perform many different functions. Some of these functions include inserting or replacing genes, deleting or rearranging genes, activating genes, and other types of gene modification. This shows the true broad reach that CRISPR can have. Although there are many benefits to CRISPR technology, there are some safety concerns as well. In particular, scientists were worried about the fact that CRISPR has no off switches to prevent it from making cuts in the wrong places. However, this concern is rapidly diminishing, as scientists have found an anti-CRISPR gene. An anti-CRISPR gene was found by biochemists at the University of Toronto. These anti-CRISPR genes inhibit CRISPR technology so that scientists do not have to worry about CRISPR going out of control. Omar?
As Eleanor and Kad mentioned, CRISPR definitely has promise in curing numerous genetic diseases. However, when using it for gene therapy, there's an essential question: price. According to Troy and Brennan from CVS Caremark and James Wilson from the University of Pennsylvania, one-time treatment gene editing therapy will cost in excess of $1 million. This colossal price tag seems enough to immediately rule out the commercial viability of gene therapy. However, it could actually end up being less expensive than current treatments. Present treatments appear comparably small at around, for example, $200,000 a year for hemophilia, but this adds up quickly. MIT Technology Review confirms this, as seen in their data, where diseases such as bubble boy and hemophilia cost $4 to $6 million over a lifetime, compared to gene therapy's predicted cost of $1 million. Although still a great sum, gene therapy would be much less expensive in the long run as it produces lifelong cures. However, there is still difficulty in how the average patient could afford its 7-figure price tag. They obviously can't afford it themselves, so health insurance companies become the only option. But insurance companies can't pay thousands of large $1 million payments all at once. Brennan and Wilson from the University of Pennsylvania offer a solution. They pay per performance model. Under this method, the large one-time payment is spread over a period of time to around $150,000 per year. and successful performance acts as reopeners to the annual fee. This improves treatment quality and relieves financial stress. However, this solution doesn't come without its problems. There can be difficulty in defining successful performance. For example, if Glyberia, a treatment for high triglycerides, showed a reduction in biomarkers or chemical improvement, but showed no reduction in frequency of hospitalization or physical improvement, then the treatment may not be considered successful. Circumstances like these complicate the pay-performance model. But there's also a solution: patient service teams. These teams monitor behavior past drug usage to ensure a more holistic evaluation of treatment success, though they may most likely raise treatment costs slightly due to an increased staff size. Nevertheless, the pay-performance model, coupled with these patient service teams, remains the best solution for patients to pay for gene therapy. CRISPR definitely has its benefits both medically and economically. However, some say this technology gives too much power to scientists, especially if used on germlines.
Germline editing is the modification of reproductive cells, or in other words, altering human embryos. A survey done by the University of Wisconsin and Temple University in Pennsylvania found that when it comes to using gene editing in order to enhance germlines, just 26% of the sample population found it acceptable, while over 50% found it unacceptable. On the other hand, if gene editing were to be used exclusively on somatic or non-reproductive cells, only 35% objected. From this, it is obvious people don't necessarily find gene editing itself atrocious, but rather despise the idea of creating designer babies. After all, human embryos aren't some kind of customizable teddy bears, customized to anyone's desires. This idea is absurd. To further demonstrate the morality of germline editing, a study done in 39 countries by Motika Araki and Tetsuya Ishii of Kaido University found that out of these 39 countries, 75% of them, the ones shown here in pink, have all banned it. These include Canada, United Kingdom, Spain, Australia, etc. It is obvious many countries believe germline editing is unacceptable. If germline editing were to become legal, people would seek to enhance the same traits in their children, such as eye color, attractability, intelligence, etc. If this were to become a reality, Marcy Luvansky of San Francisco Center of Genetics and Society predicts the world will face inequality like never before between designers and non-designers. Designers who possess better qualities in every aspect compared to those who are non-designer, either because they could not afford the huge price tag Omar previously mentioned, or because they deem it immoral. In no time, designers will be seen as normal and even superior to those who are non-designer, who would be marginalized as genetically inferior outcasts of society. Introducing such a thing would create a massive social gap within developed and especially developing countries. Simply put, this line should not cross. It has never been okay to play the role of God. However, this system can be completely avoided if restricted to only somatic cells, providing cures to an unimaginable number of diseases, as Eleanor and Kat stated.
Thank you, Noah. The extensive reach of CRISPR technology provides many benefits that enable it to cure a vast majority of genetic diseases in the human population. Despite all the significant ethical concerns Noah mentioned and the economic complications Omar expounded, the medical potential of CRISPR is incredible, surpassing other gene editing technologies in the past, as Eleanor stated. The pay-per-performance model, although limited in defining success, will offer the solution to allow commercialization of gene therapy. One solution would be to ban gene editing altogether. However, this is too detrimental, as it would be almost like rejecting the cure to cancer. Instead, to reap the maximum benefits from this technology, we should prohibit enhancing germlines while continuing to edit somatic cells to cure diseases. Although this might be somewhat complicated, as we would have to cure each generation instead of just the parent cell, and although the legislation required to accomplish this may be complicated, it is definitely plausible and definitely worth the risks. The gene editing revolution has just begun. Continuous research and experimentation with this technology will allow us to refine it and help save millions of lives across the globe. Thank you.
Thanks. Okay, we'll start with you, Omar. Can you, what was the strongest counter-argument to the solution your team identified, and why was it a good one?
Great question. I think the strongest counter-argument was probably Noah's perspective about how it can actually be very unethical to edit somatic germlines. For example, like that could lead to people, like he said, changing intelligence, and that if that means like our solution, if it wasn't strict, then it could lead to all of that inequality.
Great, thank you. All right, Kat, in the future, what change would you make to your group norms, and how would you expect that to improve your team's presentation?
In the future, I would make us collaborate more, because we collaborated throughout the presentation, but it would really help if we collaborated right at the beginning. And I think that, like Omar, he took initiative and helped us work out our ideas together. But if we had really got together and started right at the beginning, it would have benefited us more.
All right, thank you. No, could you, in what way, what is a way in which your team's resolution makes you think differently about your individual research?
Well, in my individual research, I was strictly con. I only did research on the con side of journal editing, but we just in our solution we decided to. I thought gene editing altogether was very detrimental and that it really didn't have any benefits. But looking at Kat and Eleanor's solution and how they gave examples of how it actually cured individuals with certain diseases, I figured that there is a different perspective to gene editing, and that really made me change my thoughts about it.
All right, thank you. And Eleanor, what is an example of a compelling argument from one of their research papers that you decided to exclude from the presentation?
Well, Kat's original scientific research talked about CRISPR and how it may not work in all cases, but we found that information was actually outdated, and new recent information proved that was untrue. So we decided to take that out.
All right, thank you guys. Great job.